Hello everyone, welcome to today's hearing. And today's hearing we are going to talk about acute inflammation, but we are going to base on cellular events. In our previous hearing we talked about acute inflammation and particularly vascular events. Now today we are going to talk about cellular events. So in our previous video we talked about the blood vessels, we talked about the tissue, we talked about the infectious organism releasing neurotoxins and destroying the tissues over here, phospholipid being converted into arachidonic acid, and are going to find ways to release some substances. Some of these substances were the interleukins, the leukotrienes, the prostaglandins, and some histamines. We also talk about bradykinin. So we also talk about the increase in vascular permeability, whereby we say that there is increase in vascular permeability when the endothelial cells contract, leaving behind some gaps. So this causes some plasma fluid to leak out leak out from the vasculature. Now, these substances, when they bind to receptors on the endothelial cells, these are the receptors on the endothelial cells. When these cells contract, apart from contracting to increase vascular permeability, they expose substructures known as selectin. So selectin traps some immune cells, for example, neutrophils, monocytes, trap some immune cells. As they trap, the immune cells try to uncoil as they travel through the vasculature, and that end up the immune cells end up leaking through the gaps which were created. This process is known as diapedesis. The process is known as diapedesis, whereby immune cells leak from the vasculature through the gaps into the interstitial fluid. So some of these immune cells which leak out, we are going to see some of them as we have the neutrophils. We also have some macrophages. So these are the immune cells which leak out through the gaps. We have neutrophils, we also have macrophages. So what happens? The neutrophils and the macrophages tries to clear out the bacteria. It's the bacteria. So the neutrophils and the macrophages try to clear out the bacteria. So what happens? The immune cells are attracted towards the source of infection, that is the bacteria. So when the immune cells are attracted towards the source of infection, where the bacteria is, let's say we have the neutrophil there, we have the bacteria here. We have the bacteria. We have the neutrophil which has been attracted to where the bacteria is. So what happens? The neutrophil forms some pseudopods to try to bring the bacteria in towards itself. So the neutrophil forms the pseudopods to try to bring in the bacteria towards itself. So this process is known as phagocytosis. The process is known as phagocytosis, whereby the neutrophils try to get some pseudopods so that they can bring in the bacteria. We have the macrophages. The macrophages also does the same thing. We have the bacteria here. So the macrophages try to place some pseudopods to try to bring the bacteria in towards the cell. So the process is known as phagocytosis. So in the process of phagocytosis, the bacteria end up being in vesicles. So, so this is the neutrophil. We have the bacteria here. We have the neutrophil and the bacteria. Phagocytosis has taken place. The bacteria has been engulfed. It ends up in vesicles. So the net results will be this way. The end result will be that way. So we have the neutrophil trying to secrete some pseudopods, trying to engulf the pathogen. So the pathogen has been engulfed. Here the end result is this one. We have the neutrophil, we have the pathogen which has been engulfed into vesicle. So what do we call this one? We call this one a phagosome. A phagosome is a vesicle in which the bacteria has been enclosed in. It's known as a kind of phagosome. Then the neutrophil, we have some lysosomes here. Lysosomes. So what happens next? Next thing, we have two vesicles. The vesicle containing some lysosomes. We have another vesicle containing phagosome. So the next thing, the vesicle containing lysosome fused with the phagosome. These are neutrophil. So here we have the lysosome fusing with the phagosome. Now, this one is what is called phagolysosome. We call this one phagolysosome, where the lysosome is fused with the phagosome. We call it phagolysosome. Then what happens next? We know lysosome has what? The lytic enzymes. The lysosome contain lytic enzyme. The role of this lytic enzyme is to hydrolyze or to break that down any foreign organism. So lytic enzyme from the lysosome destroy the bacteria in the phagolysosome. So we have the bacteria. So we have the fusion of phagosome and lysosome forming phagolysosome. The bacteria within it has been hydrolyzed, has been broken down. Next thing, we have the phagolysosome now coming towards the membrane. The phagolysosome comes now towards the membrane. So we'll have another structure. So this is a neutrophil. So when the phagolysosome comes towards the membrane, what happens? It expels its contents outside. So this process is known as exocytosis. This process is known as exocytosis, whereby the contents of the phagolysosome are expelled outwards. The process is known as exocytosis. So the exocytosis materials, that is the bacterial antigens, are not taken through the lymphatic system to the nearest lymph nodes. And that's how some of it is clear. Now let us have a look at what happens in the macrophage. So this is the macrophage. Now let's have a look at what happens in the macrophage. So with the macrophage, this is one of the antigen presenting cells. We have three antigen presenting cells, the APCs, we have the macrophage, we have the B cells, and the dendritic cells. These are called the professional antigen presenting cells. So because we looked at the three antigen presenting cells and we realized that macrophage is one of the antigen presenting cells, what happens? With the macrophage, the cell process happens. The phagocytosis, formation of phago lysosome. But now because this one is an antigen presenting cell, it has something else. So it's a macrophage, but again, it contains something. What this structure known as the MHC2. So this is the MHC2. Once the bacteria has been engulfed, it undergoes process. So it is processed, and then the bacterial antigens are now presented. So this is the bacterial antigen. The bacterial antigen is now presented through the MHC2. Once it is presented, what happens? It is also now taken to the nearest lymph node. We'll get to know in the next video what happens when the macrophage containing the bacterial antigen on the surface, what happens to the nearest lymph node. We'll get to know what happens there in the next video. So thank you very much. Today's CME was talking about cellular events, and we've seen what happens once there is leakage of the immune system into the interstitial space. Thank you. Watch the next video, we'll get to know about the MSC2 and the macrophage. Thank you.